the room here, but we're going to get started on our introduction. Welcome to the Midwest CASC's October Science Seminar. We're going to be showcasing some of our graduate student research projects today. For those of you who haven't been here before, I've got a couple housekeeping tips before we get started. One is that this webinar is being recorded, so please mute yourself until we have our Q&A time, and you can choose whether to have your video on or off during presentations. Another note is that one of our presenters sadly was not able to join us today. She had something come up at the last minute. So we only have two presenters today, which means that they get to take a little extra time, and you all should take a little extra time to think of questions to ask them. But it's likely that we are going to wrap up a little bit early before the end of the hour today. And again, welcome to the Midwest Climate Adaptation Science Center's October Science Seminar. I'm going to hand things over to my colleague, Alyssa Welch.
Thanks, Jess. I am Alyssa Welch. I am the program manager for the Midwest CASC based at the University of Minnesota. Let's see, Jess covered this slide here. So before we turn it over to our speakers, I am going to give just a very brief introduction about the Midwest CASC. Again, many of you are very familiar with Midwest CASC. Some of you are joining for the first time. Welcome. And we just want to stress here again that the Midwest CASC is a partnership for regional research collaboration. The Midwest CASC comprises 8 institutions that you see on the map here, and we are hosted at the University of Minnesota. We partner with USGS to conduct climate adaptation research, and we're part of a national network of regional CASCs that work on similar issues. Each of us have our own regional science priorities that guide our work that are relevant to issues affecting our region. The CASCs overall work to produce science that seeks to inform resource management and help fish, wildlife, water, land, and people adapt to a changing climate. And let's see. For the Midwest, advance a slide. I'm not sure if I want to advance a slide. Let's see. Oh, we'll go back.
No, we just
All right, we'll go back. So at the Midwest, we accomplish this research in a couple different ways. First, we conduct what's called synthesis research on relevant regional priorities, and I'll talk a little bit more about that in a little bit. We support a network of graduate students and postdoc fellows, 2 of which are joining us here today to share their research. We also partner with our consortium member, the College of Menominee Nation, to offer a tribal undergraduate research program, or REU program, for the summer. The program has been going on for 2 years here, and we'll be going into our 3rd year coming up, and we're starting planning for that. We host capacity building workshops and an annual gathering for the Midwest community. Some of you may have joined us for our August gathering in Indianapolis. And TBD on timing and location for our 2024 gathering. We are working on that now and hope to be announcing that soon. And very importantly, we fund through USGS climate adaptation research projects and partnerships. So that is a key function that the collaboration serves. So moving into our announcements for today, an important part of the synthesis research that I mentioned earlier is that there are postdocs centered at each, there will be postdocs centered at each of our consortium member institutions that will help to shape and conduct climate-related research on these synthesis projects over the next few years. And we are excited to share that University of Wisconsin and Indiana University have posted their postdoc announcements. So please head to our website, check those out, share with your relevant networks, and watch for a few others that will be posted in the coming months. We are also excited to be heading to Duluth. There are a group of us from the Midwest CASC heading up to Duluth this week for the Midwest Climate Resilience Conference. We will be hosting several sessions and an exhibitor booth, so stop by, connect with us, say hi, and watch for more announcements in the newsletter about where we will be across the region. So Jess, I'll turn it back to you to introduce our speakers.
Yeah, thank you, Alyssa. And I realized I didn't introduce myself at the top of our seminar here. I am Jess Delfiaco. I'm the communications manager for the Midwest CASC. And as Alyssa mentioned, we do support a network of postdoctoral researchers and graduate students throughout our region at our various member institutions. And these students and postdocs work on climate adaptation research, of course, which you will hear more about shortly. And we also offer them training sessions, networking opportunities, and other ways to engage with the climate adaptation research community in our region and share their work through presentations like on today's seminar, lightning talks and a poster session at our annual gathering, and blogs on the Midwest CASC website. So if you are looking to learn more about what our postdocs and students are up to, there's several ways you can learn about that. We actually just kicked off this year's early career programming, our training session series just a couple weeks ago. And we are excited to be planning 4 different training sessions between now and the spring. And these focus, each one will focus on different aspects of the climate adaptation cycle and will feature expert presenters and different training focuses. But we're excited to have the group kind of set up for the year here. But without further ado, I want to welcome our speakers for today. Joe Young is a PhD student at the Michigan State University, and Miao is a PhD student at the University of Minnesota. We're going to start off with Joe, and before I hand things over to him, I did want to say that rather than waiting until the very end of the hour for our Q&A session, I'm going to invite everyone to ask questions of our presenters after each presentation. So we'll take a few minutes. You can talk to the presenter, and then of course we'll have extra time at the very end if you have any additional questions. But yeah, get those questions ready and be ready to ask them at the end of the presentation. With that, I'm going to stop sharing, and Joe, you are welcome to pull up your slides.
Okay, sounds good. Hey, we're seeing everything properly? Okay, good.
That's good.
Yeah, so my work focuses on the ecophysiology of jack pine throughout the Great Lakes region, but specifically with special interest in Michigan for some reasons that we'll get into. And it's also in partnership with a lot of different institutions and things. Once again, we'll get into a little bit about why that's important. So just the real kind of fundamentals behind what I'm looking at. So post-logging era, they basically just raised the entire state of Michigan. So you had all these species now that were trying to bounce back, not just plant and tree species, but also bird species, like specifically the Kirtland's warbler, which is a neotropical migratory bird, which is seasonally endemic to Michigan, at least now it is. So it only has its breeding range within Michigan, and it's directly dependent on these jack pine trees. Jack pine are very fire prone and things. So post-logging era, when there was all of this fire moving through the state throughout the 1800s and things, jack pine was something we didn't really want a lot of on the landscape. So between fire suppression as well as just not planting it and favoring red pine for timber, it really reduced the population of this bird, so much so that it was one of the first things listed as endangered. So there was a Kirtland's warbler rehabilitation scheme that was developed and implemented starting in the 60s. And then in 2019, the bird was delisted as endangered, but is still very much conservation reliant. So part of the framework that was established to keep the Kirtland's warbler on the landscape uses this adaptive management framework, at least in more recent times. And it works with primarily the Michigan DNR and the U.S. Forest Service, as well as a number of other parties. And so they kind of describe themselves using this schematic that's on the right. So you have the conservation team, which is nested within the broader conservation community. And then there's a number of different subcommittees and working groups therein. I specifically am in the Habitat Improvement Working Group. So we are able to communicate directly with land managers in the DNR and the Forest Service about what seems to be going on the landscape and what they're not understanding. We're able to research it and give them updates on how things are going and help them implement it. Also, more broadly, because we're working in the southern range of jack pine, and we're looking at climate adaptation specifically, this has become a pretty significant interest to Ontario, which has a lot of jack pine on their landscape. And, as things warm up, they're going to see a climate more similar to what we've experienced now. So this data could also ultimately be very useful for them. Again, getting into that kind of rationale. So as this schematic was originally set up, the trees were going to be harvested about every 50 years. Started in the 60s, and a lot of those original plantations now are getting to the point where they're supposed to be harvested. But it's really apparent that these trees are not worth a lot. And the way that it's supposed to work is the revenue generated by the harvest of these trees would go back into planting and further conservation work for the bird. So they've been considering extending the rotation length out to 70 years, but most of the projections of jack pine growth, not just in Michigan, but throughout the US, are really extrapolating on the younger sort of age structures of jack pine. So in mature stands, we don't really know a huge amount about how they're going to continue growing, if they're going to sort of plateau. And then also to complicate things, we have the climate change of it all, which we don't know how exactly the climate is going to influence jack pine, or at least in what magnitudes. So kind of the working model that we have here for what we think is going on in jack pine, and this is built on a lot of fundamental plant physiology and theory, a few citations in the bottom there. So the atmosphere is always thirsty for water. And a huge part of how that influences trees is that the trees are taking up water from the soil and it's going through the trunk, and then the atmosphere, based on how thirsty it is pulling it back out of the stomata of the trees. And the rate at which the trees are able to keep up with that is mediated by some of the physical characteristics of these trees. So that's things like how wide the stem is, how big the cells are, but then also importantly, how tall the tree is. You can kind of think about it like if you have a really long tube, there's more physical resistance stopping the movement of a fluid through that tube than a really short tube where it only has to travel a small amount of distance. So that creates kind of a sticky situation, being that we're looking at these big mature trees and we're trying to get more trees to that point. But those are the trees that are going to be most likely to experience resistance and trying to keep up with atmospheric draw. So as things get warmer and the atmosphere gets thirstier, those large trees are at the highest risk of mortality and reductions in productivity and things like that. So based on that kind of fundamental model, the idea would be that trees growing on warmer sites are going to plateau more quickly than trees growing on cooler sites. So while they're young and very similar, they're not going to experience that much resistance, but those on warmer sites with a warmer atmosphere struggling to keep up with the atmospheric draw are really going to become limited and be able to put on less and less wood every year. And also it just helps them limit the actual physical resistance to keep up with that atmosphere. For those ones on cooler sites, they're not going to be coming across those sort of climatological and physical limits quite so quickly. So our sampling design, we started off using just Michigan and eventually expanded into Ontario to get a larger climate gradient. But you can see Michigan on the top right there is way down in the very southern edge of that range of jack pine, which is kind of like that dark green color. So this all occurred on Michigan DNR land, as well as on Ontario Ministry of Natural Resources land in jack pine that's 70 years old and older. 70 years, again, is kind of what they've been tossing around as maybe extending rotation length to. So if we look at those trees and we can reconstruct their growth pattern, we'll know all the way up till their planned harvest how those trees are on average growing. And with this large latitudinal gradient, we're able to see about 5 degrees Celsius of temperature variation from our southernmost trees all the way up to our northernmost trees. So getting into the actual field methods, we start out and we go into some of these stands at predetermined plots, and we will just do a standardized plot inventory for everything standing within a roughly 10th acre plot. It's about it's 400 square meters. So every tree gets a diameter and a height, as well as a live crown ratio and the height at 3 inches, which is an important number for land managers because that tells them how much actual wood volume you're going to be able to get out of these trees. Another thing that we did was we did a little bit of investigatory work to find out the rate of decomposition for logs on the ground. That way we can use logs on the ground as well as standing dead trees to figure out what proportion of all these trees that we're seeing on the landscape are dying. And we want to make sure that it's recent mortality, not mortality from long ago during stem exclusion and things like that. So we were able to identify that about that level 3 of decomposition correlates with roughly about the last 20 years. So if we were only using 1, 2, and 3, we can be pretty sure that those trees have died within this period of climate and not from historic stem exclusion and things like that. So once we've conducted this whole plot inventory, we choose one dominant and one co-dominant tree from each site. And then we take 9 different samples out of those trees. So those are cross-sectional samples. I'll call them cookies. And we take one at the very ground to get a total biological age of this tree, a breast site cookie, just because that's kind of standard for dendrochronological work and things, as well as a percentage of different heights, all the way up from 15 to 95% of total height of these trees. That ensures that every single tree is getting the same number of samples out of it, where instead of just using every meter or so, different heights trees would have a different amount of data available. Each of those cookies then are sanded all the way up to 600 grit, so we can look under a microscope and with a high level of confidence be sure where each one of the growth rings is. So then every cookie, we know how high on the tree it came from, as well as how many rings are on it, and that tells us how many years ago the tree reached that height at which the sample was taken. So you can see on the left side here, that's the tree on the ground. We're taking these cookies out of it, and those cookies just all stacked up. I always think they look kind of funny to reduce a tree to this little pyramid. But then you can see that plot in the center. Each one of those points is an individual cookie. So it has an age known as well as a height on the tree known. And then we're able to fit that function above for every site level to find out how those sites on average are growing. So using that information and using that function that's determined to be unique for every single site, we can find out at age 50 how these stands are all going to compare. If we compare it against ages, then that becomes kind of a confounding factor as the trees get older and taller. So we want to all measure them at a uniform age. So we use 50, which tends to be about standard in North America. In Europe, a lot of times they'll use 100. So what we found, though, is that height at age 50 on the y-axis and the x-axis is increasing temperature from left to right. As temperature increases, we are seeing shorter trees by age 50. So that speaks to the idea that as trees are struggling to keep up with the atmospheric draw for water, they aren't growing quite as much. And then this also includes some information about our different types of soil that we're seeing on the landscape. By far, it tends to be very sandy soil. Some have a little bit of clay in them. And then up in Canada, you get these sites that have a little bit more of that sort of coarse till, and you reach a lot of large stone relatively quickly. And we can see that when you have a little bit more water holding capacity in those cases, like the sandy loam or in the coarse till, that has a positive effect on this relationship. But overall, still pretty negative relationship between how warm it is, as well as how tall those trees end up being. Another question, of course, then was about mortality. So what we're seeing here again on the x-axis is temperature increasing from left to right, and then the proportion of trees that are living on the y-axis. So that's of all the trees on the landscape that were measured within our plots, which proportion of those were living on a given site. And again, we're seeing that as things get warmer, fewer and fewer trees have remained living. So we're seeing more and more mortality, or at least more proportion of death as that temperature increases. Once again, that little bit of clay in the soil and that little bit of coarse till does have a positive impact on survivorship among these trees. So if we think back to that original hypothesis, however, we're measuring these single points in time, age 50 or just current mortality, things like that. But we're also kind of interested in how the growth rates compare and when is that plateau happening. So if we look on the right-hand side there, we're seeing curves by broad geographic region. So I just kind of call them climatological climatological regions, all of those trees clustered together tend to experience pretty similar climates. So we have a few from the northern lower peninsula, then a cluster from the upper peninsula, as well as a couple of clusters up in Canada. And interestingly, what we're seeing is that those trees, which are experiencing more heat and likely more stress, actually maintain a fairly linear growth rate over time, where the trees that are on cooler sites, which we would expect to fare better over time, are actually, in some cases, plateauing earlier. So much so that if we were to take those curves and project them out, in the long term, we would see the ones on cooler sites being shorter than the ones on warmer sites. It's just that the ones on the cooler sites seem to be initially benefiting right from the get-go from that cooler climate. So we're kind of thinking about this and wondering what could be causing this. So currently, the theory that we have working is that there might be sort of a relationship between that mortality that we're seeing and how that frees up soil water and other nutrient availability. And there could be kind of a compensatory relationship between mortality and resource availability, which means they're just barely able to continue growing on those warmer sites. Where on the northern sites, which are cooler, and we're not experiencing all that mortality, they could be hitting a wall harder because there's more competition going on for all those resources above and below ground. So now we're kind of entering into the realm of speculation, next steps. So everything that we went up to until now, that was all funded by CASC and the DNR, and that was 1 year of funding. And then now this is work that I've begun on the second year of funding. And so first we're trying to understand how trees die, which is kind of an old question, but still one we don't have a great answer to. And so there's kind of two leading schools of how trees die, one of which is carbon starvation, which we see here on the right. So we can see that the heights might align relatively closely, but if we look at the actual radial growth of these trees, we're seeing that trees that ultimately die, you can detect quite early. They're not putting on as much new wood radially every year as those that stay living. Again, keep in mind that how much water the tree can move up to the canopy is mediated based on the width of the tube, the width of the trunk of the tree itself. So this is what we would expect to see under carbon starvation, where the stomata are just always closed, they're not fixing new carbon, and eventually they just die out of lack of resources. So these are set up to be very similar plots. So the top is the height relationship, the bottoms are the radial growth. Again, each one of those points is just a cookie. Hopefully you can kind of imagine the lines that would follow those. So what we're seeing for here in Canada is that the heights are very similar, whether or not the tree lives or dies. But in terms of radial growth, on average, all those trees that end up dying tended to be smaller than the trees that remained living in terms of the actual radius. So those trees seemed that they were dying from suppression and carbon starvation and things like that. What gets a little more interesting is when we move into Michigan. And what we're finding from Michigan, and these happen to be in the Upper Peninsula, but these trends hold true throughout Michigan, with the exception of just some odd ones, as we're seeing nearly the opposite relationship, that the trees that are dying are actually the widest trees, like they've been growing radially the most quickly. So the height stays the same, but they're growing really effectively in terms of radial width. You'd think that they're able to keep up moving all that water into the canopy, and they should be able to keep up with that atmospheric draw. So thinking about this and reading some of the literature, you find that there's this anatomical trade-off that goes into how trees actually make cells, how trees are using what resources are available to them, and what long-term sort of ramifications that can have. So we see this anatomical trade-off between implosion safety as well as hydraulic efficiency. So to be more efficient, you want wide, big, open cells that can transport a lot of water. The issue there is you can get air into it and you get an embolism, just as you see in mammalian biology or anything with a vascular system, really. When you get a bubble in the vascular system, it breaks the flow, causes problems. That means that that part of the vascular system is no longer reaching its end goal. On the other side of that, where we really want to make sure that we're not getting any air into the vascular system, we tend to see trees with small cells, with really thick cell walls. So there's this kind of trade-off between, are we going to move a lot of water up to the top of the canopy, or are we afraid of seeing embolisms occur? And I'm hesitant to call it a decision, but that's something that the tree allocates resources for differently, based on genetic influences, as well as just the historic environment it's been in. So this looks a little bit more in detail at the cells themselves. And what we see is that, so for trees that are growing radially more quickly, and the pits, which are the part that actually connects each cell to cell and makes that tube all the way from the bottom of the tree to the top of the tree and allows it to transport water, the trees are growing more quickly, have big, wide open pits. So that confirms that those trees that are growing quickly are probably trying to maximize the amount of water that they can move up. And a number of different studies have corroborated this finding, but that also, again, exposes them to quite a lot of risk. So if you are experiencing a lot of tension on the water, if the atmosphere is still really thirsty and the roots just aren't keeping up with taking in enough water, if the soil's really dry, like a drought or something, there's a lot of tension on the water, and those are the trees that are at highest risk for experiencing an embolism. So this could be part of what we're seeing in Michigan, is that idea that mortality might be punctuated by an increase in more severe climate events. So in Michigan, where our droughts are more common and more severe, they're going to be dying due to just poor anatomy, where in Canada, where those events aren't occurring quite so much, that seems not to be the case. So knowing all of this will give us insights into, maybe we can core some trees, find out how old they are, what their growth rate's been, and then on average for a site, what's the risk of mortality here, given some probability of experiencing droughts of a certain level of severity with the next so many years. And so we're collecting a lot of climatological data and using historical data to compare what sorts of droughts have punctuated these mortality events, as well as what sort of climate regimes might be influencing the trees to allocate carbon in one way versus another. And then land managers will hopefully be able to use those sort of insights to actually make changes on the landscapes about which trees we can leave to grow, which ones should be harvested. And that way we can maximize our yield and increase revenue to maintain the sustainability of this conservation scheme for the Kirtland's warbler. So again, we're not through the full year of CASC's funding yet, so I still have a lot of work to do, kind of in these two different parts. So that first part, again, we're going to be looking at the shapes of the curves. So rather than just punctuated like 50 years old, we want to find out how the shape of the curve is influenced by different parameters within the climate, the soil, and the site itself. And then part two of that is just understanding in more detail, or confirming rather, why the trees that we're seeing dying are dying and why they're different between our different sort of climate areas. So that's going to be with cell anatomy measurements and measurements of non-structural carbon allocation. Again, if trees are really stressed, they're making sort of decisions about what they're going to put all their money into investing in the future. So non-structural carbon might be something that they're not going to want to put on or put a lot of money into because it doesn't have high reward for maintaining photosynthesis in the future. Same with radial growth. And then we're also going to try to understand how those changes in carbon allocation are actually influencing the hydraulic sustainability and mobility within the trees. So you're able to put samples of wood in a centrifuge and find out when they're losing their ability to conduct water based on how hard that water is being sucked out of the tree, how that relates to the actual anatomy of the wood. Then the last part of this is using an isotope analysis. So understanding for any given tree, now that we have kind of an age and a height relationship, how we can reconstruct whether the stomata are open or closed and what it's taking out of the atmosphere throughout the course of the tree's age. And that can be extended up to the site level. Yeah, so if anybody has any questions, that's all I've got.
Thank you, Joe. Folks, feel free to ask questions by putting them in the chat, or you can go off mute, go on video to ask your question directly. We'll take a few minutes.
Yeah, is that a question from Gary?
Yeah, Joe, thanks. Nice job. Your presentation got me wondering, in Michigan, at least on state land, almost every acre of jack pine is planted.
Mhm.
And we probably have been selecting for the seedlings that grow the fastest and are the most robust over time. And it's almost like, have we set up our jack pine forest for failure under this rapidly changing climate? And I just wonder if you got any thoughts on that.
Yeah, so one, I know there's other people in our department who are interested in the relationships between this wood anatomy and the genetics of the trees. So if we are using the same seed sources with a particular predisposure of genetics to have a particular anatomy, that relationship would be important to understand. The second part of that, though, is that within jack pine management, although the DNR does an enormous amount of planting, the Forest Service tends to try to use more natural regen if possible. So oftentimes the Forest Service will leave tops on the ground and things like that, and sometimes we'll even burn a roller chain over that, and then let the cones open up and germinate, and then they'll supplement in planting to get the density required by Kirtland's warbler. The other thing is that the DNR is currently experimenting with, because we have this kind of undesirable age structure of jack pine right now. A lot of them, we have a pretty homogeneous age structure where historically it would have been pretty heterogeneous. So they've been looking at non-commercial restarts of these trees and different ways to just, on a budget, get them to restart the stand, mimic fire, without having to necessarily put fire back on the landscape. So they're looking at mastication. And with that, the whole tree stays there. It's just mulched, cones hit the ground. They scarify to expose bare mineral soil. And then the same trees that were standing there before that weren't necessarily planted or could have been natural origin, those genetics hopefully will still be maintained on the landscape and still be able to be part of the gene pool.
Yeah. So just so I make sure I heard you correctly, and I'm going to way simplify this compared to what you just presented, but if you grow fast, you're riskier than if you grow a little slower.
In terms of radial growth, yes.
Yeah. Okay, great. Thank you. Nice job, Joe.
Thank you.
We've got time for a couple more. If anybody else has a question they want to put to Joe.
Yeah, do we have another one from Brent?
Yeah, good job, Joe. So
Yeah.
you started off talking about part of the interest being the shifting age at which these trees are becoming marketable or merchantable. Wonder real briefly, what are those main market uses? Why is it shifting? And is there any potential change there? Is there a different outlet, different market that might not be as, dependent upon what you've been examining?
Yeah, so a lot of this would go to pulp for like biofuels and cogeneration energy, but the wood chip markets really started to collapse in that area. And the way jack pine grow, they're very unlikely to ever reach a size or a shape that's desirable for like commercial timber. So we've kind of been forced into this situation where there's not, the number of buyers are decreasing, and we're not seeing even the wood have reached the level of value that we hoped it would. So that's why we're trying to explore these sort of alternatives.
All right, I'm not seeing any other hands right now. So Miao, I'm going to invite you to start your presentation. And then if anyone thinks of a question for Joe, you can pop your hand up at the very end of the seminar and we'll have a little bit more time. So, all right, go ahead.
Hello, can you see my screen?
Yep, looks good.
Great. Pull out my presentation mode and my laser pen. Okay. Hello everyone. My name is Miao, and I'm a first-year PhD student in Ecology, Evolution, and Behavior program at College of Biological Science in University of Minnesota, Twin Cities. And today I'm going to present you the research project I did when I was doing my master's program at Utrecht University, which is a program called Environmental Biology. And I'm now, as I told you before, at University of Minnesota, and I'm also a member or a scholar, a student at Midwest CASC, Climate Adaptation Science Centers. And okay, then the topic of the project is called Nitrogen Enrichment Alters the Drivers of Grassland Overyielding. And I will explain to you in detail, like what's grassland overyielding and what does the effect of the nitrogen enrichment do to this grassland overyielding. And I have a lot of collaborators, as I show here in the first slide, that I would like to introduce you to, like briefly. Miro and Katie and Yang, they are from Utrecht. They are my co-advisors when I was doing my project here. And Eric, Sorinas, and Noemi, they are from University of Bonn when we are collaborating. And now Sorina is together with me doing a postdoc at University of Minnesota in in Elizabeth and Eric Siegler's group. And Dylan, Noemi is on WSL in Switzerland, doing her second postdoc as well. Dylan Craven, he is in Czech Republic right now, together with Yuri and Voltek and Yang. And Forrest Spell is my supervisors in University of Minnesota, and as most of you already know, Mao is also in University of Minnesota. He's doing he's doing his independent research in Cedar Creek. And Norman is in what's the island around Australia? I am bad at geology, as you can clearly tell. Cecilia is on Brazil now. Laser is also in Brazil, and when we are collaborating, she's on also in Swiss. Peter Reich, when we are collaborating, he's still in Minnesota, and now he's in University of Michigan. And Christy Anner, she is in IDIF, which is German Research Institute, Biodiversity Institute. And they are all of the very lovely collaborators of this project, and I really want to take some time to introduce them one by one. And I hope you can have chances to meet them or collaborate with them, and I'm happy to connect you with them if you want. Okay, started to introduce my project. As most of you already know, biodiversity sadly in decline across the globe due to a lot of climate change factors, including nitrogen enrichment. And if we don't do some conservation effect on them, they will continue to decrease. And then in the recent future, we will have like left with Earth, and Earth that has very limited biodiversity, which most of us would not hope to see. Due to a general positive relationship between biodiversity and ecosystem functionings, in short BEF, if we continue to lose biodiversity, most of the ecosystem functions or ecosystem services that we highly depending on will be lost as well, including pollination, decomposition, or biomass production, which is a very important sources of the agricultural production. And this biodiversity ecosystem functioning relationship, in short BEF, which I will call BEF in the later of this presentation, is very closely regulated by environment, including climate, land use, soil type, and pollution. And one of the most well-studied DEF that is also the focus of this introduction, this presentation, is called overyielding. So overyielding in general could be quantified by this net biodiversity effect, which I will explain how to calculate it in a few slides later. And there are two drivers of this net biodiversity effect, which is called complementary effect or selection effect, in short CE and SE. And complementary effect indicates in general how different plant species collaborate with each other in taking up the nutrients. And selection effect, as you can see here, it indicates how dominant a single species are when they are growing in the mixture comparing to monoculture. A mixture indicates a lot of species, plant species growing together. And monoculture literally means one single species growing by themselves alone. Previous studies, previous long-term grassland diversity experiments have already shown that with the increase of year, this overyielding, which is calculated, quantified by net biodiversity effect, will increase through time. And this temporal trend of the overyielding is usually due to the increase of the complementary effect with the slightly decrease of the selection effect with the increase of time. So in short, with the increase of time, a larger increase of the complementary effect with a smaller decrease of the selection effect will contribute to an increase of the net biodiversity effect, as previous studies showed. However, nitrogen availability, as I told you before, may explain the variations in this BEF relationship, especially between how many plant species are there in the mixture and how well this diversity effect created by the plant mixture. So because these nutrient additions can modify the underlying ecological interactions in a community, we could see with the increase of the nutrient, complementary effect, will decrease with the increase of the plant species richness. And the selection effect will increase with the nutrient addition. So in short, fertilizations will decrease or will reduce how collaborative these plant species with among each other, and increase the dominance of one single or a few specific species in the community when they are taken the nutrients. So in practical, this could be due to several reasons. For example, first of all, the plant species in the community, they might not have the necessity to collaborate because they have already enough nutrients to take by themselves. And secondly, there could be a competition exclusion of a specific species that are more favorable for a higher nitrogen environment, which increase the selection effect here. So, but go back to the summary table, which is the hypothesis table here. With the increase of the nitrogen amount, complementary effect will decrease, selection effect will increase, and this complementary or opposite directions of the complementary effect and selection effect will contribute. Sorry, Siri. Will contribute to non-significant changes of the net biodiversity effect, which is in general overyielding. And this has been showed in one of the previous meta-analysis that Dylan Craven, one of my collaborators, has published in 2016. But he doesn't know, what he doesn't study is, in reality, we have a lot of different nutrient enrichment way. This different enrichment way of the nitrogen will usually accumulate it in the ecosystem and across the regions and across time, and increasing accumulation of the nitrogen with continuous depositions and fertilizations could lead to an exaggerated impact on overyielding and in different regions. And so my research questions overall is, how does this overyielding and different drivers of overyielding will respond to nitrogen accumulation. And how I'm going to explore this is aggregate the effect of time and effect of the nitrogen amount to explore how does overyielding and different components of overyielding respond to this nutrient accumulation, which I will quantify with cumulative nitrogen addition. I will explain this to you in a minute. So in order to solve these research questions, I use 15 experiments that manipulate biodiversity and nitrogen addition across 10 countries in different regions, different continents. And in total, I will compile a lot of data points, which include species distribution data and species level biomass, which I will use to calculate overyielding. And the reasons why I only have these 15 experiments is because when I want to calculate overyielding, I would need to have the monoculture planted in the experiment. And a lot of the other biodiversity experiments, they don't have the monoculture, like separated plant, besides all those mixtures of different biodiversity gradient. And with all those data, I mainly use the mixed effect models in R and RStudio to calculate analyze the result and my models. And among all those experiments, I want to highlight this point in North American, which is Cedar Creek data. And I use biocom experiments in Cedar Creek, which has 23 years data until the years that I conduct this analysis. But now we have more data available, which I really recommend you to check out. It's really amazing. So before we go to the result, I want to briefly explain to you how I calculate the 3 components of overyielding so that you can have a brief concept of how these different components of overyielding represent what kind of mechanisms behind overyielding. So for example, when first of all, let's look at the monoculture biomass here. For example, if we have 4 different species, which was represented by 4 different colors here, and the different species, they have different monoculture biomass, which means they produce a certain biomass when they are growing alone. And when we plant these 4 species together in a mixture, theoretically, these 4 species will just have 1/4 of their monoculture biomass and to compose of the mixture biomass. So if we grow these 4 species together, we will have 1/4 of this dark purple block and 1/4 of this blue and green and yellow block to compose of, to make up of the mixture biomass if there's no overyielding. So this is a non-hypothesis of this no overyielding happens. And then the mixture biomass can be equal to what we expected from monoculture biomass. However, if there is one species or a multiple species that are more dominant or that are producing more biomass comparing to what was expected from their monoculture, for example, the purple species here, it should be in theoretical 1/4, but it actually produces more than 1/4 when they are growing with other species. Then we say there is a positive selection effect happening in here. And however, the other scenario is all species are producing more biomass in mixture when they are growing together, comparing to when they are growing alone, which is the monoculture. You can see from the table, the plot I highlight here, that all of the species, at least 4 colors, they are producing more than 1/4 of their original monoculture biomass. Then we say here is a positive complementary effect. However, what I want to highlight here is you could tell this yellow cute plot here is the worst productive in monoculture, but they are this yellow block is the most productive species in monoculture in mixture. Sorry, so this is what we call negative selection effect. I will repeat that again. The worst performance in monoculture turn out to be most productive in the mixture. Then we call there is a negative selection effect. And this negative selection effect or positive selection effect can happens at the same time with complementary effects. So they are not exclusively exclusively happening. And that's what we call three components of the overyielding. And usually the net biodiversity effect could be summarized by the additions of complementary effect and selection effect. Okay, I know you are already done with the math. And we will go to results for now. Firstly, we show the relationship between species richness and three components of the overyielding, including net biodiversity effect, complementary effect, and selection effect here. And the solid line here means the relationship under the ambient conditions, and the dashed line here represents the condition scenarios of what we after we add fertilizations, and different colors represent different studies. And as you can see, with the increase of the species richness, complementary effect, as we expected, decrease. Selection effect, as we expected, increase, but still a little slightly negative. And this opposite and proportionate decrease of complementary effect and increase of the selection effect add up to contribute to a non-significant changes in the relationship between species richness and net biodiversity effect. In short, nitrogen addition does not alter the overall overyielding because of this contrasting impact of the complementary effect and selection effect. Secondly, we assess whether or not this nitrogen impact depending on the amount of the nitrogen we add into the plot. So with the increase of the nitrogen amount, whether or not the net biodiversity effect and complementary effect or selection effect changes. And as we expected, again, complementary effect decrease have a larger decrease with larger nitrogen amount that we added into the plot, and selection effect have a larger increase with increasing nitrogen amount. And this contrasting effect of the complementary effect and selection effect again contribute to the non-significant changes of the nitrogen net biodiversity effect, which is the overall overyielding. However, we would really love to see whether this nitrogen addition effect are affecting the overyielding by a binary scale, which is addition or not, or a continuous scale, which is how much we add. So we separate the data set into including ambient, which is including the nitrogen addition equals 0 point, which is the ambient point, ambient condition, and exclude ambient, which is only increasing the nitrogen amount with those plots that have nutrient addition. And we interestingly, we found when we take out the ambient conditions, there is no significant relationship between increasing nitrogen amount to different components of overyielding. I know these figures is a little bit complicated, so I put it in a simplified version in next slide, which means overyielding responses only depending on whether this community receives nitrogen or not, regardless of how much nitrogen we add into the plot. I know this is a little bit counterintuitive, also to us, also to our collaborators. So we take a step further to see whether, going back to our general research questions, whether this non-detectable impact of nitrogen amount, whether is it result from the non-linearity effect of the nutrient accumulation across time, and turn out it does. So this is the result the main result of our project, which is the increase, the relationship with cumulative nitrogen effect, which is the multiply of time, the year of when we put nutrient into the plot, multiplied by how much nitrogen we put into the plot every year. And we found the nitrogen accumulations decrease the complementary effect very soon after we add nutrients into the plot across time. And this quick decrease of the complementary effect lead to the net biodiversity effect in decrease when we have lower level of nutrient accumulation. However, with continuously increase of the nitrogen accumulation, the selection effect continues to increase, and then the slope of the increase also. And this increase of the selection effect actually lead to the bounce back of the net biodiversity effect with higher level of nutrient accumulation. And this inflation point, which is the turning point, happens around 40 kilograms per hectares per year. Not per year, sorry. It's a total amount of nutrient. So when we in total add less than 40 kilograms per hectare, the net biodiversity effect, which is the overyielding, will decrease. However, if we continue to add more than 40 kilograms per hectare of the nutrient into nitrogen into the plot, then we will see a bounce back of the net biodiversity effect. And for your information, the nitrogen deposition rate in the Netherlands, which is the place when I conduct this research, is 50 kilograms per hectare. And the usage, according to the Netherlands, the Dutch Agriculture Department, the limit of the usage standards of fertilizations in the plot is 230 to 250 kilograms per hectare per year. It's not even the total amount, it's per year. So you can see how desperate or how alarming the Netherlands are facing the nitrogen crisis at that time. And I want to put out this nitrogen deposition global map here again, which I have already shown you during the introduction. Let how many regions have already have the dark colors more than 40 kilograms per hectare per year. This is also a figures in 2008, and the nitrogen deposition rate is also higher in some of the regions in 2023 right now. And yeah, I just want to remind you of that, the alarming situations we are facing now in face of the nitrogen crisis. So take home message here. First of all, the amount of the nitrogen addition does not alter overyielding because of these contrasting impacts on complementary effect and selection effect, which means we can fortunately have a lot of diversity effect along a wide range of the fertilization gradient. And second of all, overyielding responses depending on the nitrogen accumulation in the plot is instead of the nitrogen amount per year that we add per year or time. And this diversity effect might disappear with even higher nitrogen accumulation since the selection effect cannot increase unstoppably. The extreme condition is that there is only one plant species in the plot, and then this one single plant species will dominate the plot, and then there's generally no net biodiversity effect at all. And I would love to talk about this like later in the Q&A if I didn't explain it clearly enough. And the second of all is we need to be more cautious about the co-effect of the atmospheric depositions and fertilizations. And yeah, that's all of my presentation. I know I ran a little bit out of time, and I want to shout out for my group in the orchard and with my advisor Yang here. And if you have any questions, I'm happy to stay longer.
Thank you, Miao, and thank you, Joe.